Before starting this module, I will brief about some previous ingredient and the requirement of the fermentation media on industrial level. Up till now, we have discussed about carbon source, nitrogen source, phosphorus source, and other different minerals. Along with these requirements, we talk about some buffers, precursors, inducers, promoters. So these are the different ingredients that directly or indirectly affect on the growth of that organism, or directly or indirectly on the production of our desired product. But in this module, we will talk about the oxygen requirement, and then some about the trace element and the pH and osmolality of the medium. In detail, we will check about, throw a light on oxygen requirement. Oxygen requirement, as we have already discussed, that is only need when the organism has to grow aerobically. But in case of those organisms which have not to grow in the presence of oxygen, we have to deplete the oxygen from the medium by using different reducing agent, oxygen reducing agent, or some oxygen chelating compounds. But when we are dealing with the oxygen requirement, then there is different aspects which we have to be very critical while making the medium, because different media component have different effect on the oxygen solubility. Normally when we say that when there is a water and nothing is added into that, that is a specific oxygen soluble capacity. So as well as we add different ingredient into that, and the concentration of the medium is going on, then the solubility of the oxygen decrease accordingly. So as well as the solution will be more concentrated, then there is the less oxygen solubility. So during the fermentation process, what happens when there is the utilization of the ingredient, on the other hand, there is the increase of the biomass, so the need of the oxygen change. But there are different factors that affect on the oxygen requirement. The first of all, the fast metabolism. So as well as the fermentation process will going on, and then there will be the more cell inside the fermentation process, then ultimately that can lead to the increase in the metabolism rate. So the culture may become the oxygen limited, because as well as there will be the more cells, then there will be the most viscosity. And that ultimately what happens, then the oxygen solubility will be decreased. So then the starved conditions is produced that effect on the oxygen requirement. So that depend upon directly on the oxygen demand. So then there will be a more oxygen demand in that conditions. On the other hand, when we say about the rheology, so the individual component of the medium can affect on the viscosity of the final medium concentration. And after all, if we increase that, and just to increase the aeration and agitation, just to meet the oxygen requirement, then that also affect on other in term of foam formation. So on the other hand, this is also directly linked when many of the antifoams we use just to decrease the foam production, then that also affect negatively for the solubility or the dissolved oxygen inside the fermentation medium. So the fast metabolism, rheology, and the antifoamers that affect on the oxygen deficiency in the medium. That's why there will be a more oxygen requirement during the fermentation process. So while making the medium, we should select those which have less interference with the oxygen solubility while talking about aerobic fermentations. As concerned the trace element, so just keep in mind, we have discussed some minerals in our previous module when we talk about the mineral part of the medium. So some minerals are required in a bulk amount, but some are required in a very trace amount. So as we already know that the trace elements are mostly present along with the carbon, nitrogen and phosphorus sources, but sometimes when we are dealing with the pure compounds, then we have to add the trace amount separately into the fermentation medium. You can see in this slide there are the number of trace elements that include, you can see here. So as concerned the pH, so we also have some idea when we were talking about the buffers and their use in fermentation media. The main object of the buffers and a buffers compound additionally are supplementation of those buffer compound into the media, that just to maintain the pH. So at that time in that module we discussed about the addition of calcium carbonate and some phosphate compound that regulate the pH in the medium. But during the fermentation process, the pH can be controlled by the addition of some alkali and the acids just to maintain the pH during the fermentation process. So these are the different. So continuous pH control is achieved by the addition of sodium bicarbonate or sodium hydroxide with fast mixing, because when we have to add some alkali or the acid into the fermentation media, just we have to quickly diffuse that feeding point. Otherwise, that can affect on the microorganism present in that particular site. So, as concerned the osmolality of the medium, that is specific to dealing with some specific organisms in case of bacteria. So, we have to keep the constant, the osmolarity that is needed by that organism. We have to keep the osmotic level of some specific salt to maintain the cellular level constant. So there are different amount of sodium chloride we add, as we know that by making the medium of LB medium or nutrient broth, we have to add some salt just to maintain the osmolarity of the medium. But in terms of when we are dealing with the halophilic organism, we have to just kept the high salt concentration, because those microorganisms which love to grow on high concentration of the salt, then we have to maintain the osmolarity on a high level.